What's up? What's up? What's going on, guys? 1 minute. Good morning. Time to make some money, guys. I think today I'm going to do a little bit technical analysis of bionanogenomics because right now the stock is it's kind of taking a beating, okay? I don't expect us to get to $7, so we're going to see. We will see. I mean, not only BNGO, opened with a cap down, there are some other stocks that I am watching right now that are also down at the moment. We will see. Um, so I think the webinar is going to start now. Okay. The presentation will begin shortly. I need to share this video with I have to share it online so that everyone can know that I am live. It's crazy. The market is open right now and it's very hot. I could be trading and making some money right now, guys. Join me live. The presentation is going to begin shortly. So, what am I doing? Bingo. What's going on, guys? What's up, everyone? So we are just waiting for the webinar to start. I'm going to move this to my second screen so that's okay. And I will move this one here. Okay, good. Optical genome mapping. Optical genome mapping. What's up? What's up? Where is everybody? You guys are busy making money, all right? Everyone is busy making money. Bionanogenomics is going crazy right now, guys. I mean, the stock is kind of taking a beating right now. I don't expect us to get to $7. That is where I am drawing my support. I really don't know what is going on with the price action right now. What are you guys doing? The presentation will begin shortly. So I'm waiting for this presentation of unrivaled structural variant detection for cancer. And today is going to be, the presenters are Dr. Yannick Delpo and Dr. Darin. I think Dr. Yannick is working for Bionanogenomics, and Dr. Darin is more or less from Sanford Bonham Pre-Based Medical Discovery Institute. So these two guys are going to be talking about what the Bionanogenomics Sapphire is able to do, guys. So if you can hear me, please drop a comment. Let me know, guys, if you can hear me. Let me know if the sound system is working fine. Mr. Invest is writing with me right now. So, while let me know if you can hear me in the comment section, guys. Let me know so that I can do a little bit technical analysis of Bionanogenomics stock. It seems the stock is kind of taking a beating right now. Let me know if you can hear me, guys. Market is open. I have some stocks I am watching right now. The market is open. So, um
by the sports viewing experience, and then it becomes much more than a one-way experience. It becomes a two-way experience, and that's the future that we have imagined. That's actually the future that we were looking to capture with our partnership with Sinclair, and we're really excited to prove that out. And actually, Arizona is going to be a place where we will especially focus, because again, it's a place where we do have the online access and the sports net, so it's like that perfect opportunity. So look, we're just so excited that Jason saw the opportunity with us. Obviously, he had choices with other partners. We're excited that the governor of the state said, "Hey, you're right. There's no reason why a women's professional franchise shouldn't have the access that we just gave to these 6 other men's, professional sporting events." And from our perspective, it's going to be a way to introduce, sports bettors to women's sports, but also, women's sports audience, sports betting. So it's just a win-win all around.
Given where we are, from where you sit, given where we are in the pandemic, are you seeing pretty crowded casinos and crowded facilities right now?
Oh yeah. I mean, it depends, state by state. Different states have been opening up at different times, and I think that there's sort of different levels of open. But for the most part, the physical casino business has been great. I think that just people have been for a long time, with more limited choices as to what they can do with their time. They've had essentially forced savings, and now they're just starting to have some time to go out and enjoy. So yeah, no, it's been we've seen great business across all of our casinos, and we're excited for that to continue on through the rest of the year.
And Jason, before I let you go, I'd be remiss if I didn't ask. How you feeling? Like tonight, like are we gonna do this thing? How in the last couple of weeks been for you guys on the Sun side? It has been a wild ride, and look, as far as I sit here today, a couple of injuries on the Eastern Conference side, I feel like you guys have got to be the favorites right now.
I had a feeling I'm going to get asked about the Suns as well, which is great. Obviously, we're this morning keenly focused on this deal with Bally's and promoting women's sports. But yeah, look, it's been an exciting run for us on the Sun side, and been a very competitive very competitive series.
Bionanogenomics.
A lot of fun to watch, and it goes right back to the issue of fan engagement. If you give people a product that's interesting to watch, they're going to tune in, and I think at times sports betting into that, you can just imagine how much that's going to amplify it. But we're looking to go out to LA and close it out tonight, and hopefully we can do that. So exciting times for both of our franchises.
Oh, Miles, you can go get off now. Go place your bet.
I got there's 12 hours to tip off, so I got a lot of work to do before that.
All right, well, fair enough, and congrats to you both on the deal. Valley's Chairman Sue Kim and the CEO of the Phoenix Mercury and Phoenix Suns, Jason Rowley. Good to see you both.
Thank you, bud.
All right, straight ahead, I found one stock market measure that is screaming stocks are overvalued. More on that one coming right up.
What's up? What's up? What's going on, guys? What's up? What's up, guys? I think this webinar is going to begin in the next 5 minutes, okay? In the next 5 minutes. 5 more minutes, they are going to start the webinar. For those of you wondering what I am watching right now, this is what I am watching right now, okay? Bionanogenomics is taking beating right now, guys. I mean, hopefully we're not going to get to the $7 level, okay? If we get to that $7 level and we break it, you guys should expect the price of BNGO to come down. You should expect the price to come down. So, Taiwo 366. Good morning, brother. Good morning. What's up? How are you? New Intelia, Virgin Galactic, CRISPR. What's going on, guys? What's up? The webinar is going to begin shortly, so I'm just here, like, chilling. The webinar is going to begin shortly. I think in the next 2 minutes it should begin. Sela, BBB. BTNR. No.
Hello, everyone.
Oh, okay.
Section for Cancer Research and Precision Medicine. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by BioNanoGenomics. To learn more, visit bionanogenomics.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. All questions will be answered via email following today's presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Dr. Yannick Delpou, Head of Clinical Affairs, BioNanoGenomics, and Dr. Darren Ben Finlay, Scientific Director, Tumor Analysis Research Assistant Professor, Stanford Burnham Prebys, Medical Discovery Institute. Dr. Delpou, you may now begin your presentation.
So, this is it, guys.
Hello, everyone. I'm Yannick Delpou, and it's my pleasure to welcome you to this BioNano workshop session to discuss what optical mapping can offer for the study and the characterization of cancer sample. QR codes will be displayed during this presentation to take you to relevant publications or webinars, so you might want to have your smartphone ready. Among all biological tissues, cancer is probably the most complicated to interrogate. Genome instability constitutes a major challenge for the characterization of such a sample by current technologies. And add to that the high cellular heterogeneity, the presence of many clones bearing different genetic alteration. This explains the limitation of NGS and how it led to a lack of understanding of the role of structural variation in cancer, which remains largely overlooked today. And that is very easy to understand when you take into account that 2/3 of the human genome is repetitive, making it extremely challenging for algorithms to reassemble the reads in these regions. And the consequence is that many structural variants will remain invisible to these technologies. Getting reads of several dozens of kilobases using long-read sequencing is an improvement for sure, but when you deal with heterogeneous samples, the coverage is key. When you want to address highly heterogeneous tumors with a highly clonal population, what's the point of spending tens of thousands of dollars in sequencing if you just get a fraction of the information you seek? The way our technology works isn't based on sequencing and can be compared to karyotyping, but at a much higher resolution. With karyotyping, what you have on the left, chromosomes are stained in a way you can distinguish a pattern of bands that is unique throughout the genome, with a band roughly every 5 megabases or so. And any abnormality in this labeling pattern would indicate the presence of a chromosomal alteration. The same way, with optical genome mapping, a pattern of labels is applied throughout the genome, this time with one label every 5 kb on average, and will be scanned and analyzed automatically by the Sapphire system. And similarly to karyotyping, any abnormality in this labeling pattern would indicate the presence of a chromosomal alteration. Here, there is no culturing, no amplification, no denaturation, no fishbone needed. And this is how it works. On the left part of the screen, you can see that electrophoretic cycles are applied to the DNA that will be linearized and elongated in hundreds of thousands of nanochannels in parallel. And that allows the acquisition of a massive amount of data. By running the instrument a bit longer, you can increase the yield to coverage levels that are simply out of reach of any long-read sequencing technology. And this you can do at no extra cost. On the right panel, you can see the raw data generated with the DNA counter stain in blue and the green labels as the green dots you can see here. Another major difference with long-read sequencing is the size of the molecule we imaged. They're above 200 kb, so we even have the luxury to disregard any molecule smaller than 150 kb for our analysis. So we combine all you need to address complex cancer samples, long-read information at high coverage, and for a few $100s per sample. The workflow is extremely simple. You extract the DNA using our kits to obtain ultra-high molecular weight DNA. This DNA is labeled with our enzyme called YLE1 and counterstain, and this is where the specific labeling pattern is applied genome-wide. The stained DNA is then simply loaded on the chip where the nanochannels are engraved, and from there, everything is automatic. The DNA molecules are imaged, aligned together based on the labeling pattern to create a consensus map that we call an optical map. We also measure the coverage genome-wide to detect any large gain and losses in the genetic material. And ultimately, the sample map in blue is compared to a reference in green. This can be the adjacent tissue, a control cell line, or simply a reference like a HT19 or HE38. And it is this step of map comparison that will identify any deviation in the labeling pattern that indicates the presence of a structural variant. This is how we represent the map alignment. We always represent the reference in green and the sample map in blue. Here you can see a deletion and an insertion, and we can detect that by measuring the shrinking or the increasing in the distance between the labels compared to the reference. The same way we can detect a repeat expansion like this. We do not need the repeat unit to be labeled. We can detect duplication where this pattern of label from the reference is found duplicated here and over there. Translocation when your map aligns to two different genomic locations, and inversion. And again, we can detect any large variation in the copy number by looking at the coverage. You can visualize your cancer genome in our software called Access, and we represent the results under the form of a circus plot. But let me actually take you to the live instance of Access. You do have the cytobands on the outside corresponding to each region of each chromosome. The dots are representing structural variants that are identified by this map alignment. The color code corresponds to insertion, deletion, inversion, duplications. Copy numbers are shown here with two aneuploidies identified in this sample, and translocations are the pink lines at the center. Here you do have an intrachromosomal translocation. And every time you will click on an event, the map will open to show you the details of this structure variants. So here we do have an insertion of 6 kb that is absent from a control database that is directly embedded into our software and allow you to filter the vast majority of the polymorphic variants you can find in a sample. And these variants overlap with the CLC-A4 gene. And here are shown the different structural variation classes which are detected by optical mapping. So we just saw whole chromosome changes with monosomies and trisomies, CNVs, whether they are terminal or interstitial, duplications for which we also give you the insertion sites of the duplicated fragments and its orientation information that are missing from array approaches, insertions, inversions, pericentric and paracentric, and of course, no problem to detect balanced or unbalanced events like translocations. And we can detect these events down to 1% allele fraction thanks to our high coverage, and this automatically and genome-wide. We can detect, of course, repeat array expansion and contraction. And while we outperform NGS for the detection of structural variation, there is still some complementarity between the two approaches, as we do not provide you ATGNCs because we are not sequencing based. So single nucleotide variants and very small indels of a few base pair will be reported by sequencing. But this performance of optical mapping to detect variants missed by NGS and long-read sequencing in particular can be observed in results from a recent publication from the Human Genome SV Consortium. In a well-characterized cell line, not highly rearranged as the leukemia sample could be, for example, the sequencing-based method based on the PacBio HiFi missed about 1/4 of the variants identified by BioNano. And many of these SVs that were missed overlapped with complex regions of the genome that can cause macrodeletion or macroduplication syndromes, for example. The publication did classify some SVs as detected by these sequencing-based methods. But upon further analysis, however, most of these variants were in fact identified by optical mapping, but simply classified differently. And overall, less than 2% of the large variants detected by PacBio were actually missed by optical mapping. So we provide the entire workflow for the genome analysis of a cancer sample that includes this Sapphire Genome Imager, which will scan the DNA, the chip on which the nanochannels are engraved, the chemistry to extract and label the DNA, and of course the software, the Access software I just showed you, and that is free and that you can download directly from our website. We also provide data sets if you want to play with. But given its unique capabilities, optical mapping is playing a central role in generating results that redefine our understanding of cancer in fundamental and translational research, whether it is to study key mechanisms of carcinogenesis, to characterize cancer models, or to identify new biomarkers. And this opens new avenues for clinical applications, for the identification of actionable events, and also for the modernization of current diagnostic methods. So let's dive into what we can offer for the elucidation of fundamental questions in cancer research before moving to our value proposition in translational research and precision medicine. The major cellular pathways are known to be altered in cancer, and their understanding is necessary to identify new biomarkers and develop new drugs for personalized medicine or patient monitoring. DNA methylation is one of them and undergoes tremendous changes during carcinogenesis that entirely redesigns the gene expression profile in cancer cells. And recent work from the Tel Aviv University described a method to interrogate the methylome by differentially labeling CPGs on top of the genome-wide labeling pattern applied by the DLE enzyme that we use for SV detection. And the bottom panel describes the methylation status at the single molecule level of the 53 region, which is and its neighboring enhancers. This exciting work has potential applications, whether it is to identify new epigenetic changes and new patterns at low allele frequency in cancer for biomarker discovery, for example, to monitor the efficacy of demethylating drugs that are commonly used in leukemia treatments, and also to develop new compounds directly targeting DNA methylation. DNA replication is also a major target for a new generation of drugs in personalized medicines. And for example, being able to monitor replication for progression allows to understand the replication timing in genomes, identify fragile sites, but also assess the efficacy of drugs targeting DNA replication and DNA repair. And this can be done by incorporating fluorescent nucleotide in live cells to label replicating DNA on top of the DLE1 labeling pattern that we apply on the genome, and this to map replication fork progression genome-wide. And here, this has been used to measure the replication timing on chromosome 20, as you can see in this figure. But fundamental science is mostly about laying the foundations, the first stones in our understanding of completely uncharted territories. And extrachromosomal DNA illustrates this perfectly. It's an obscure DNA structure displaying a massive oncogene amplification, but its structure, its effect on gene regulation, and its role in cancer in general is completely unknown. And NGS alone cannot extrapolate any structure because the reads are too short, and the optical maps can be used to scaffold these reads and make sense of these NGS data. Optical mapping and the computational analysis of whole genome sequencing were used in this study in combination to demonstrate the structure and make sense of circular ecDNA sequence for the first time. Beyond these applications to study the basic mechanisms in carcinogenesis, a profound understanding of the genomic rearrangements occurring in cancer cells, whether they are study models or patient biopsies, is paramount to improve diagnostic methods, developing new drugs, and improve patient care. And BioNano excels in this domain. And let me focus now on the detection and the resolution of structural variation in cancer and how patients can benefit from it. Many cancer study models are established and characterized and associated with specific genetic defects that help to understand the developments and evolution of this cancer. But because of the limitation of NGS and cytogenetic approaches on tumor tissues, it is clear today that a lot of genetic alteration remain to be discovered, and that our understanding of these models is by far incomplete, with the certainty to identify new biomarkers or therapeutic targets. So in an effort to better characterize uveal melanoma models, the group of David Jeancien at the Curie Institute in Paris used high-C RNA sequencing and optical mapping to identify structural variation leading to fusion genes. And over 1,000 translocations were predicted by high-C data in the two models they used, MP41 and MP46. But only 50 could be cross-referenced with their whole genome sequencing data, and none could be validated down the road, illustrating the issue of false positive calling and the burden of data creation when working from sequencing data. The RNA sequencing approach is pretty heavy. It's identified over 300 fusions using a combination of 5 algorithms and extensive data creation. And only those called by at least 2 algorithms and in 2 independent replicates were considered solid. 14 were kept for validation, all of them found by optical genome mapping, without the need of a complex bioinformatic pipeline here. And they could be further characterized. I think this example really illustrates the simplicity of optical mapping and how we can tackle the identification of fusions from the DNA while providing more information than RNA sequencing. So here, optical mapping detected a tandem duplication at this locus. You have this sequence here in the reference that actually aligns in the sample map over here, but also over there. This leading to a specific fusion between the ACAT10 gene fusing with the MPK gene over there. This was independently identified by optical mapping from the DNA, confirmed the fusion detected by RNA sequencing, but on top of the fusion detection, optical mapping provided the mechanism leading to this event, which is a tremendously important information. So the take-home message here is that you can identify any fusion in a genome without information needed about the fusion partner, and you can interrogate any region, such as IGH, which is notoriously difficult to deal with by RNA sequencing. And this, even if your event is present at low allele fraction, because we provide a coverage to do so. But now I would like to switch gear and look at more complex variants. As I mentioned earlier, our conception of the role and nature of structural variation is largely biased due to the technical limitation of NGS technologies. Beyond simple variants like deletion, duplication, translocation, inversions, complex structural variation like chromothripsis, chromoplexy, or templated insertion chains, TIC, can cause multiple DNA junction in distinct topologies. And by sequencing thousands of cancer genomes, new complex SV patterns have been identified, but resolving their exact structure is not always possible from NGS data. You can see here a structure called Pyrgo. Pyrgo means tower in Greek, and it corresponds to a low junction copy number hotspots. It has been identified specifically in patients with ovarian cancer. You have at the bottom evidence from the NGS data of duplication, but the structure of the event cannot be deducted. And this nested structure, you can see here, was resolved thanks to optical genome mapping. One allele is actually bearing all the alteration in the background of a normal allele with a copy number of 5. Analyzing these patterns of duplication with optical mapping is essential because it reports not only on the contiguity of the loci, but also the order of the fragments. So by combining optical mapping and NGS, you can reconstruct extremely complex events. And it is important as these new SV classes can cluster tumors and identify subgroups of patients associated with a DNA repair defect and poor prognosis. And this, of course, paves the way to improvements in patient management. I just mentioned TIC, so templated insertion chains. It is a complex SV class that happens to be associated with a specific signature in hepatocellular carcinoma. You can trace back this signature to patients that develop this liver cancer due to an infection by hepatitis B virus that integrates near a cyclin gene. And this causes a complete deregulation of the cell cycle and replication stress. But resolving TIC from NGS data alone isn't possible because the reads are too short. All you can do is make hypothesis with 2 different models here, whether it is a chromosome 3 segment from here inserted into a chromosome 1, or a chromosome 1 segment inserted into a chromosome 3. And because optical mapping can completely reconstruct the region with single molecules spanning hundreds of kb, you can unambiguously resolve this event and see here that it is here in the sample map, a piece of chromosome 1 here that is spurred in the context of a chromosome 3, validating the second possibility. So identifying simple and complex structural variation is always a challenge with NGS, as we just saw, but the determination of a complex structural signature for patient stratification takes it to another level. And in the same cohorts, the automatically generated BNGO results were sufficient to cluster patients based on the presence of replication stress due to the deregulation of the cell cycle following HPV infection. And these patients harbored a lot more chromosomal instability, and you can see at the center the tremendous amounts of interchromosomal translocation in addition to tandem duplication and insertion that are reported here, slightly differently from the plots that we generate. And this cluster is different from the patient without this HPV insertion, where these patients show way less chromosomal instability. And this has exciting potential in terms of patient stratification and identification of subgroups that can correspond to a particular, that can respond, to a particular therapy like PARP inhibitors. And again, this doesn't require a complex bioinformatic pipeline. The circus plots that are automatically generated allow you to cluster these patients. The same way in head and neck cancer, HPV insertion can cause extensive chromosomal rearrangement as well. And the circus plots can cluster patients into two groups. The patient with an episomal form, so not integrated, that will display minimal level of chromosomal instability, and those with an integrated form that will harbor a tremendous amount of recombination and translocation, as you can see. And from these signature obtained from our map alignments, Dr. James Broach reconstituted the complex events triggered by HPV insertion represented on these cartoons. You have here in one sample this HPV insertion that will trigger a duplication with HPV junction between the duplicated fragments. In this sample, it will trigger a triplication of this fragment over there, but it can also lead to more complex rearrangement with here a translocation between chromosome 11 and 8 with HPV at the fusion between these two chromosomes, and the most extensive rearrangement here with a focal amplification of that region mediated by HPV, followed by a duplication of these neighboring fragments. So the take-home message here is that having single molecules of hundreds of kbs to entirely reconstitute highly rearranged loci opens the possibility completely out of reach of long-read sequencing, whether it is to make sense of NGS data, to find new SVs or biomarkers, or to assemble complex structural signature. But how does it apply to the field of precision medicine? This is what we will discuss in the last part of this workshop. And on that note, I would like to hand it over to Dr. Darren Finlay. Dr. Finlay is a research associate professor and director of tumor analysis at Southern Burnham Presbyterian in La Jolla, California. He is a cell biologist with a broad interest in oncology and drug sensitivities, and has been stratifying responsive patient-derived cancers based on genomic features, including structural variants using the Sapphire system. So Dr. Finlay, the floor is yours.
That introduction, as Yannick said, I'm Dr. Darren Finlay, and I'm from Sanford Burnham Prentice, and my talk today is going to be talking about the use of optical genome mapping to uncover novel structural variants in leukemias, and how we can associate these structural variants with drug sensitivities. These are my disclosures. So just a brief introduction to acute myeloid leukemia, which is the main focus of my presentation today. Leukemias in general are actually increasing in prevalence over the last 20 years, which is probably due to an aging population. And acute myeloid leukemia is not a very prevalent disease. There's only about 21 to 22,000 new cases a year in the United States. However, it is a major killer. more than 50% of those people will die. So some patients do respond to standard chemotherapy, which is cytarabine, a DNA analog, and also a alkylating agent, which is usually a member of the rubicins, daunorubicin. I explicitly mention that because I'm going to show an example of that later. Some patients do respond to the standard of care, somewhere 30 to 35%. However, they almost all relapse. And the only cure for acute myeloid leukemia is bone marrow transplantation where possible. So there's a major unmet need to identify new or extant drugs, but also to identify patient populations that will be responsive to these drugs. So for our preliminary studies, we have analyzed 9 samples. We've actually analyzed more than 20 now, but our initial studies is just going to be focusing on these 9. This is the basic clinical information. What I really want to highlight is that through traditional cytogenetics, either karyotyping or FISH, we know of 6 of these structural variants that are already present. That is the deletion of chromosome 7 in this sample, BCR-ABL translocation in these 2 samples. This sample also has a deletion of the Y chromosome. And this sample has a rearrangement of the CBFP gene on chromosome 16, which is a known event in AML. Finally, this one has a deletion in chromosome 6. What I really also want to point out is that 4 of these samples are considered normal or negative by traditional cytogenetic analysis. Traditional analysis will find nothing wrong with these samples, even though they're obviously not normal because they do have leukemia. So it was of interest to us to see what could we find using optical genome mapping. So this here on the left-hand side is the concordance between the traditional karyotyping FISH cytogenetics and what was found by optical genome mapping. And all of the events I described in the previous slide were detected. That is the deletion in chromosome 7, this BCR-ABL translocation from chromosome 9 to 22 in these 2 samples, the deletion of the Y chromosome, the inversion event on chromosome 16, and this deletion on chromosome 6. But on the right-hand side here, what you can see is that there are many, many more structural variants that were also detected, even in the samples that were considered normal by traditional cytogenetics. So we usually range from around 40 up to as many as 80. And all of these structural variants are filtered against a database of healthy volunteers. So none of these structural variants have ever been found in a healthy individual. These are all almost certainly involved in the pathogenesis of the disease itself. And the color scheme, again, is going to be consistent throughout. We find mostly deletions and insertions, but we do find duplications, inversions, and some translocations. This is just one example from one of our relapse samples. So we knew when this patient first presented that they had this deletion on chromosome 7. You can see it here in the gene track. We can also see many more deletions across chromosome 7, which could well be involved in the disease progression. But we also detected 2 translocations that were not found on the initial presentation. That is this chromosome 2 to 11 translocation and 5 to 11. So this suggests that traditional cytogenetics is not sensitive enough to detect these, or that these translocations were present in a very low clonal population that is now taken over, suggesting that optical genome mapping could also be used for progression, to track progression of the disease. And just one last example of a circus plot. This is one of our samples that was considered normal or negative by traditional cytogenetics. And we don't see any translocations here, but we do see all the dots around here in the circus plot represent structural variants. We found 13 insertions, 37 deletions, an inversion, and 2 duplications. So a lot more structural variants detected using optical genome mapping. And indeed, one insertion event that we detected in this sample, and insertion events, I'm led to believe, are extremely hard to detect using traditional next-generation sequencing methods. But here we find a 32 kilobase pair insertion in the middle of a lysine methyltransferase 2A gene. And this gene has been implicated in AML before. So a sample that was considered normal by cytogenetics, we find this potentially pathogenic insertion in a known disease-causing gene. So we do have 100% concordance, but this table here is supposed to show that we've detected a lot more structural variants, and we have manually curated what we have found to try and associate them with genes that may be involved in cancer progression. So you can scan along the gene list here and find your favorite gene. The real point of this is to show that we detect all the different kinds of structural variants, and the color scheme I'm using throughout is going to be consistent. What we find is we mostly detect deletions and insertions. We also find duplications, inversions, translocations. And just the two examples I'm going to show up the top here, BCR, able. It's a translocation. We find it in 2 of our samples, but it's always the same thing. For example, we always, BCR able, it's always a translocation. This here in CDK11A, it's always a deletion. The 2 samples have it as a deletion. Similar here, it's always the same. But these are probably just not random events that we're detecting. These are probably true causative events that are involved in the pathogenesis of this disease. So that is one of our data sets, structural variants, but what we're also interested in is differential chemical sensitivity. So all the samples that we take, we screen them against 120 FDA-approved oncology drugs, and we decide their viability. So this here shows the differential chemosensitivity. That is, we're only showing the drugs that actually show sensitivities that are different in at least one of our samples. I'm just going to highlight some of the drugs that I'm going to use as examples later on. The BCL family inhibitors, which are now used in leukemia. Venetoclax is approved. Topoisomerase inhibitors, such as doxorubicin and valrubicin, which again, as I said, are frontline agents in leukemia. Another example I'm going to use is dasatinib, or targeted kinase inhibitors, which are not used right now in leukemia, but are used in other blood cancers. So this volcano plot here is the integration of these 2 diverse data sets. Every spot in this volcano plot represents a structural variant that occurs in at least 2 of our samples that is associated with a change in drug viability. That is in blue, structural variants that are associated with resistance to the drugs in question, and in red, structural variants that are associated with sensitivity to the drug in question. So as I said, every spot here represents one of those associations. I've just highlighted a few here that I'm going to give as specific examples in the next few slides. But what this shows is that there's a wealth of information you can get from these 2 diverse data sets once they're overlapped. This is just another way of showing our top associations. In this waterfall plot here, we see the top 5 structural variant drug associations that sensitize to the drugs. On the other side, the top 5 structural variants that are associated with resistance. As you can see, is that BCR-able translocations are involved in sensitization to some drugs, but resistance to others. So we chose this BCR-able fusion as our proof of concept. And these are just the 2 circus plots here at the bottom, showing this translocation from chromosome 22 to 9 in both samples. If you remember from the start, one of the samples also had a deletion of the Y chromosome, and that's marked here with the arrow. And the lower panel here is just the genome level map. So we can see the optical genome map in blue. It's tracking along chromosome 9 perfectly until it gets to this breakpoint in the middle of the able gene. And then it maps back up again on chromosome 22 in the middle of BCR. And what we find is these 2 samples, when compared to our other 7, are much more sensitive to the targeted kinase inhibitors, dasatinib, nilotinib, bosutinib. This is not a surprise, because these agents are known to target able and similar kinases. And in fact, imatinib, or Gleevec, was actually designed as a selective inhibitor of BCR-able fusion. So this is not a surprise. This is our proof of concept. So what this shows us is that we can, by using an unbiased manner, we can detect structural variants, we can detect drug sensitivities, we can overlay these, and we can find associations that we would expect. But what of associations that we would not expect? Thankfully, in this sample, the BCR-able, the 2 samples that are BCR-able translocation are more resistant to BCL2 family inhibitors. In this case, an experimental agent, ABT737. But as I said, other agents, such as venetoclax, which target the same proteins, are now used for leukemia patients. So patients that do have BCR-able translocations are less likely to respond to this drug based on our findings. Similarly, with the proteasome inhibitor carfilzomib, it's not effective against the samples with the BCR-able translocation, but is effective against the wild-type samples. Another example is a gene called CDK11A. It's also known as CDC2L2 tumor suppressor. And just one of the gene maps we're showing here shows a rather large deletion right across the body of the CDK11A gene. So this could indeed be causative or involved in the pathogenesis of this disease for these patients. Interestingly, the 2 samples that do have this deletion in the CDK11 gene are more sensitive to the rubicin family of drugs, doxorubicin, daunorubicin, valrubicin. These are all structural analogs. And as I said at the start, Unrivaled then is a frontline agent. So although CDK11 may be pathogenic for this disease, there is a good prognosis or potential prognosis value for patients that receive Unrivaled. And just again, an unbiased association, another indirect DNA damaging agent, canipicide, shows a similar profile. And then one final example is a deletion in a gene I had never heard of before, but again, it shows the value of this sort of unbiased analysis and detection. The deletion in a gene called KIF26B. This is a kinesin, which is involved in trafficking of cargoes along microtubules. So it was gratifying to see that the two samples that had a deletion in this kinesin were resistant to a microtubule targeting agent, paclitaxel. They're also resistant to mitomycin C and triethylene melamine. And I'm not really going to discuss it today, but I'm just going to allude to it. We also study other diseases other than leukemia, and in 3 of our childhood brain cancers, we have also found a deletion in KIF26B or its homologue KIF26A. So in summary, optical genome mapping shows the 100% concordance with traditional cytogenetic analysis. But as I hope I've shown, we don't just find those 6 major translocations and deletions that we knew were there, we also find many, many more. And that using these integrative analyses, we can associate these novel structural variants with chemical sensitivities and drug resistance to clinically relevant agents. And just by whet your appetite, we don't just do this in leukemia, we're also analyzing glioblastomas, childhood ependymomas, and primary CNS lymphomas. And as I mentioned previously, all 3 of the ependymomas we've screened have shown deletions in kinesin family members. And with that, I would just like to thank the FORE lab at Sanford Burnham Previs, also Junyin and Rabi Murad from the Sanford Burnham Previs Bioinformatics Core, our collaborators Jim Mason and Carol Borian, who provide us with the leukemia samples from Scripps MD Anderson, and of course all the team at Bionanogenomics who were involved with analyzing the first 6 of our samples. Thank you.
Thank you, Dr. Finlay. And these are great results and really illustrate how patient will benefit from optical mapping, whether it is to identify classical rearrangement informative for a diagnostic procedure, but to profile tumors in precision medicine, thanks to our ability to detect structure, signature, and actionable fusion. And to also complement and modernize the current standards of care to improve patient management with a more streamlined workflow that you can see on this slide. We're seeing tremendous adoption all over the world, and numerous studies are now evaluating optical mapping against the standard of care for cancer patients. I would finish this workshop with the results from 3 users that nicely illustrate how optical mapping can modernize cytogenetics, identify clinically significant variants missed by a standard of care, and push the boundaries of precision medicine. The first study is a multicentric study led by Bryn Levy from Columbia University and James Broach from Penn State University. It is a landmark study where optical mapping was compared against the standard of care in 100 AML cases previously characterized by karyotyping, FISH, and chromosomal microarray. And not only Bionano identified all the clinically relevant variants, SVs and CNVs, reported by these standard of care, but it also identified additional variants in 11% of the cases that have been missed by the routine methods. So it showed the ability to identify balanced events like balanced translocation that you can see here from the karyotype between chromosome 10 and 13 and 8 and 21 that would be represented by these lines over there and between these chromosomes here and there. But also it showed the potential to identify cryptic events that were missed by karyotyping. In this example, where a translocation 821 was identified, as you can see here, but a translocation between chromosome 4 and 5 was missed and was detected by optical mapping with a deletion of the 5q terminal region that can have a significant impact to stratify patient into in terms of risk assessments. And since optical mapping is a new technology, the authors modeled the optical mapping nomenclature on the array nomenclature described by ISCN 2016. But since microarray technology cannot detect balanced rearrangement, they have proposed a unique nomenclature using this ISCN logic that you can see over there. The conclusion of their study is that besides identifying all clinically relevant SVs and CNVs reported by the combination of the technologies that constitute their standard of care, and reporting on additional SVs missed by routine methods, optical mapping also refined the genomic structure reported by traditional cytogenetics, and identify additional clinically relevant variants in about 24% of the cases. And most importantly, in 6% of the cases of AML patients presenting a normal karyotype, optical mapping directly identified additional clinically relevant variants and or CNVs, including fusion events. So this has a clear potential to improve the patient management in the future. And optical mapping identifies way more events than classical cytogenetics because we have a better resolution and we are not targeted approach like FISH, but the amount of data generated isn't an impediment to an easy characterization of cancer sample. Some structural variants, translocations, for example, are considered hallmark for risk stratification of leukemia patients. And in a few clicks, you can highlight events reported in international guidelines published, for example, by WHO or NCCN. To do this, you can simply create a panel, a virtual panel like you have on this screen. We call it a bed file that will extract from the genome-wide interrogation of the genome all the events impacting the loci of interest that you listed in this file. And of course, this panel can be customized at will and combined to one another. So in this AML panel, you do have here reported 5q deletion. You can have a 7q deletion. You have any event impacting the KMT2A gene, any event impacting the translocation H21, leading to a RUNX1, RUNX1T1 fusion, and also inversion on the chromosome 16. You can create this panel for any cancer, any pathology that you wish. We can make one for CLL, where besides being able to assess the complexity of the karyotype directly from the circus plot, you can also directly pinpoint any event impacting the genes like Notch1, BCL2, that is evaluated for patients to orient the patient towards specific treatments, and also a deletion in the 17p, including the TP53 gene over there. But actually, let me show you how it works live on a sample. This is an ALL sample where we loaded such bed file that follows some guidelines and include gene list reporting those guidelines. And you can see that comparatively to this previous circle close I showed you, you do have an extra circle over there with blue bands, and those bands are actually the genes that I documented in this bed file. If I zoom in, one is RUNX1 over there. This is BCR, but you can add any gene you want. And so in this circles plot are reported the variants that have been identified in this sample and screened against our control database to remove the polymorphic variants that are found in the normal population. And I simply have to go to this bed file and decide to show only the SVs and CNVs that overlap with this gene list. And in just a few clicks and just a few seconds, I will have just the filtering of this genome-wide information that will allow me to focus only on the variants that are overlapping with genes that are reported to be clinically significant in this cancer. If I click on this one, this is a deletion of the EBF1 gene. So it's very easy, and we can see that the identification of hallmark variants is easy with the Access software, while a lot of new clinically relevant variants are still identified, and that well, completely unnoticed with the standard of care because they were not expected and because FISH or panel sequencing are targeted approaches. And this leads me to the second study from Children's Hospital Los Angeles, where Dr. Gordon Araka reported the identification of variants missed by standard methods and not identified by their custom gene panel, since it was not expected in this type of leukemia. And this illustrates the importance of genome-wide unbiased structural variation detection as provided by Sapphire. Dr. Raka does genetic testing of pediatric leukemia at CHLA and routinely performs a FISH, karyotyping, chromosomal microarray, and their OncoKids NGS panel. And interestingly, they routinely observe about 15% of B-ALL and 5% of AML cases where the key genetic driver remains unknown. And 8 of these negative samples were run on optical mapping to determine if it can detect clinically significant genetic drivers missed by their comprehensive molecular evaluation done on their routine workflow. And 2 cases really stand out in terms of discovery that I would like to develop a little bit. This sample is from a patient that had a poor response to treatments and developed a residual disease requiring more aggressive treatments down the road. A translocation 2.5 was identified by karyotyping, but at the time of diagnostics, they did not really understand the significance of such rearrangement. Optical mapping found that the same alteration, but could locate the breakpoint at a much higher resolution. And an interesting gene was found at the breakpoint of this translocation, the PDGFR beta. This event was later, of course, confirmed by FISH and RNA sequencing. And PDGFR beta is associated with what they call phe-like ALL, which is linked to high-risk ALL and associated to a better response to tyrosine kinase inhibitors. So this patient has been treated accordingly with imatinib or tisagenib if this translocation was better characterized at the time of diagnostic. And the interesting question is, why was it missed in the first place? First of all, the fusion of the PDGFR beta with this gene XBTBN1 has never been described in the ALL before, but only in very few cases of another hematological malignancy. Hence, it wasn't expected in this type of ALL, and this is why it was absent from their OncoKid panel. This second patient was also identified as a standard risk patient and had an even worse response to treatment than the first one and had to be involved in a CAR-T trial to get into remission, however, relapsed shortly after that. Karyotyping seemed perfectly normal, and CMA didn't report abnormalities. And to their surprise, when they ran optical mapping, it reported numerous rearrangements they were completely unaware of. And this is most likely due to the fact that leukemia cells can sometimes not grow or grow poorly depending on the cultural condition. And this really illustrates the potential of working from uncultured cells with optical mapping. And here, in this pretty complex circus plot, a 3-way rearrangement between chromosome 1, 6, and 12, leading to a fusion between MEF2D and FOXG2 was identified. MEF2D fusions define a specific subtype of BALL, and FOXG2 is a very rare partner described only once, explaining why it was not part of their NGS panel and why it was missed in their initial comprehensive testing. And this fusion was confirmed by RNA-seq. And the clinical significance is that MEF2D fusions are associated with high-risk disease and very poor outcome. explaining the aggressive cancer in this patient, and that information would have been crucial to risk stratifying this patient. So it's striking to see the discoveries made in cancer sample for which the standard of care is already focusing on the identification of structural variants. And that really highlights the importance of applying optical mapping in solid tumors, cancer types where classical cytogenetics is not applied as it is in leukemia, and where structural variations tends to be largely overlooked. And Dr. Ravinda Kohli from Augusta University evaluated optical mapping against a SNP array that is part of their routine workflow in brain tumors.
Dr. Ravinda.
Among different samples included in the pilot study, they observed 100% concordance with the SNP array, like you can see on this sample. So at the bottom, you do have the OncoScan results showing all the large gains and losses identified in this sample. And you can see how they perfectly matched the results of the BNGO whole genome CNV plot that is reported on the top panel. But many other variants, structural variants, copy number variants, many translocations, completely unsuspected, were found, and many of them impacted well-known oncogenes or tumor suppressor genes like BRAF, EGFR, or CDKN2A. And this is really illustrating the importance of looking at those structural variation and having this genome-wide analysis done at high coverage thanks to optical mapping. So their conclusion is perfectly concordant with the two previous studies we saw, with a clear potential of optical mapping to identify new, unique genetic abnormalities, new fusions without any information on the genes impacted or the fusion partner needed. Think about NTRK fusions for which dozens of fusion partners have been described. You clearly do not want to use 30 different fish probes here. And also to the potential to improve SV characterization in those complex karyotypes that have been shown by Dr. Raka. So the results are so conclusive that this study is on the path of a laboratory developed test for solid tumor at Augusta University. So as a conclusion, optical mapping is a long-range technology that overcomes NGS limitation and access regions of the genome inaccessible even to long-read sequencing, because it's due to highly repetitive sequence, because these regions are flanked by segmental duplications, or because they are composed of high GC contents. Its coverage up to 1600x coverage can interrogate heterogeneous tissues and allow the detection of structural variants down to 1% allele fraction automatically, genome-wide, and for just a few $100s per sample. Unlike RNA sequencing, it can detect fusions automatically and provide additional information on the mechanism leading to the fusion event, even if this event is falling into complex regions, such as the IGH locus that is notoriously difficult to deal with by RNA sequencing. And this without any information on the fusion partner. And of course, it will also detect any SV or CNV affecting the regulatory sequences. From the different evaluation, we saw that optical genome mapping can identify classical rearrangements, informative for patient diagnostic or risk stratification. It shows outstanding concordance with the standard of care, but at a much higher resolution. It can identify actionable, druggable events without a priori information often missed by conventional workflows. And it can also reconstruct complex structural signatures automatically without the need of a complex bioinformatic pipeline to run. So I really hope all these applications convince you to try our technology. You just have to flash this QR code to access our website and get in touch with our sales representative. And we will be happy to answer any question you have in the chat or by directly getting in touch with us. On these notes, I will thank you for your attention. I would like to thank again Dr. Finlay for being with us today and presenting such outstanding results. So until next time, goodbye.
Whoa, so it's finished, guys. I mean, that was amazing. You guys saw the conclusion where he was talking about the importance of the Sapphire device and, of course, the difference between the Sapphire device and next generation sequencing devices, such as the PacBio device, or short read sequencing device from Illumina. So I think that's where we're going to end this live stream, since I have very little viewers right now. So I'm just gonna record my video, and hopefully you guys are going to check it out. Okay, have a wonderful day, guys. See you in the next video. Ciao, ciao.